Hello everybody. How's everybody doing? Hope everybody's fine. My name is Michael, and welcome to my presentation. So over the summer, I worked on stem cells with Jane and Nandhini in the flow core, and we worked on determining cell surface markers of human-induced pluripotent stem cells. So the question we aimed to answer was, what cell surface markers are expressed on human-induced pluripotent stem cells? So first, let's start off with how we get these cells. So we use the Yamanaka strategy. So we take an adult. From an adult, we extract fibroblast cells. From there, we use transcription factors to reprogram those fibroblast cells into human-induced pluripotent stem cells. These human-induced pluripotent stem cells can be reprogrammed into two main types of cells using transcription factors and specific media. These two cells are neurons and cardiomyocytes. So from the literature, we see that the cell surface markers TRON60, SSEA3, TRON81, CDH3, SSEA4, and CD30 are all on these cells. However, this is the first time in the experiment that all those cell surface markers are in one panel. In order to answer the question, we use a flow cytometer. So the formal definition of a flow cytometer is flow cytometry is a process whereby single cells are measured for fluorescence and light scattering properties. It's also used to count the cells as well as find its relative size and check for or look for granularity of the cell. So looking at the image, the laser hits the cell, and using the Fluorescence scatter detector, we're able to find its relative size. Due to a fluorophore being attached to the cell, a light is emitted and it falls into one of the four channels. So in my experiment, we had HIPSC cell from the stem cell core. From there, we washed, added antibodies, incubated, and washed again, and used a flow cytometer and analyzed the data. So the results. So from our results, you can see that the HIPSCs express all the cell surface markers in the antibody panel. At the bottom is showing a negative control for the markers TRON81 and TRON60, just for visual reference of what an unstained panel looks like. So what's next? The floor core is now going to work with the stem cell core and groups to try quality control the HIPSCs come into Gladstone. This could include cell sorting, deposit cells with these markers, which could be much quicker way to obtain pure HIPSCs than current methods. Cardiomyocytes and neurons are the main use of reprogrammed HIPSCs at Gladstone. So the flow core would like to develop markers to test for the efficiency of the reprogrammed HIPSCs. Also, the panel will be tested on disease cell lines like cancer cell lines to see if they express these markers. With that, I would like to give a special thanks to my mentors, Jane and Nandhini. Thank you, Sudha and Emma as well, and the core lab, all of the interns that made this experience great. And thank you, PUMAS Grant.
Thank you, Michael. Any questions for Michael? Oh, I do have one question for Michael. Apologies. Did you also try any intracellular marker?
No, we didn't try any intracellular markers.
I just want to add, sorry, as Michael's mentor, we didn't try that because we're trying to keep them alive so that we can grow them further.
Perfect. Thank you, Michael and Jane.